Sorry.
Now
Hi
you start.
everybody, this is Orhan Akman. I am researcher at Columbia University. I work on adult polyglucosan body disease diagnosis and the treatment. So the title of my talk is the Glycogen Branching Enzyme Deficiency, Molecular Pathogenesis, and the Antisense Oligonucleotide Treatment for a particular mutation that we have described in 2015. This slide briefly summarizes the pathways that if something goes wrong on those causes glycogen accumulation. I mean, depending on the enzyme, of course, the degree of glycogen accumulation changes and causes the disease. I mean, based on the enzyme, each enzyme causes different kind of disease. Although they cause glycogen accumulation, the progress of the disease is different from one enzyme to another. Now, talking about the glycogen accumulation, glycogen may accumulate either as a normal glycogen or its structure may change and become a different form of molecule. And in this case, we call it polyglucosan. And there are few enzymes that causes polyglucosan formation. One is the branching enzyme, and the others are the laforin, and RBCK1, and the PFK, phosphofructokinase. So although we know the mechanism very well for the branching enzyme and the PFK deficiency, we don't know exactly why laforin or RBCK1 causes polyglucosan body and the glycogen accumulation. Now, the glycogen molecule is a form of storage molecule for the glucose. Glucose is the sugar that we get it from the food, the carbohydrates that we ingest in our meals, and after it is being digested in our system, the cells get it from the bloodstream and store it by binding them to each other. So there are two enzymes that makes this structure. One is the glycogen synthase, which binds every glucose molecule one after another using 1 and 4th carbon. And the branching enzyme removes a part of this molecule and makes the attachment back to the molecule again using carbon 1 and 6. By using this mechanism, the glycogen is being synthesized as a highly branched carbohydrate polymer. And if there's any problem in branching, or if the glycogen synthase activity is very high, then you have this molecule that you see here that I am going to talk about. It is molecular mechanism and disease in this presentation. The branches are very important because if the glucose is being synthesized and stored in this way without any branches, it looks like starch or the cellulose. And as you know, those two molecules are not really soluble, and it may cause problem in our cells, in our body, in the brain, in the neurons or muscle. It's not going to be able to use this molecule, and it is going to occupy very precious space, especially in neurons, and it causes neuropathy and the muscle disease. And of course, this is one side of it, occupying the space, and also the cells are not going to be able to produce enough energy when they need it. There's a reason that glycogen is being synthesized to be able to provide the fuel when the cell needs energy. If you have this molecule, the cell is not going to be able to get energy, and in case of patients, they are going to feel tired very quickly, and the muscles are going to break down because they cannot provide enough energy to maintain their structure. So this animation shows how the glycogen is being synthesized. As you can see, the the glycogen synthase makes the longer branches, and the branching enzyme makes the branches. And in this way, you have a long, very highly branched molecule. And I kind of ignore to mention about the starting the glycogen molecule. It starts with the glycogenin, and this is a self-glycosylating protein. It is deficiency also causes polyglucosan body disease, but the mechanism is mostly depending on the balance between glycogen synthase or the number of the molecules in our cell. And this is also cause a very rare disease. But my main topic is going to be glycogen synthase and branching enzyme in this talk. So if there is no branching, of course, there will be a very long glucose stretch. And as I mentioned earlier, since this molecule is being synthesized and used up from the ends, only single end is not going to be efficient for our cells to produce energy from the stored molecule. And of course, when the molecule become like this, it becomes insoluble and difficult to reach by enzymes. And unfortunately, in our body, there is no enzyme that can jump into this structure and cut the glycogen. And that's why this molecule sits in the cell almost forever. So what's the branching enzyme deficiency and how it is being, how it happens? So as you know, every enzyme or the protein in our body is encoded by DNA. And mutations occurs in DNA, and that reflects on the protein. And if there is a very the adverse or the strong mutations that changes the protein structure, that also affects the enzyme activity. There are certain mutations that actually totally destroys the enzymatic activity. In case of branching enzyme, if there is no branching activity at all, it is not compatible with life. So the babies are born still, and they cannot survive beyond a year of age. And if they, of course, ever born. Generally, this disease don't cause much problem in utero. Babies are born, they are alive, yet the problem is they cannot obtain the enough energy from the stored glycogen or polyglucosan in their case. So they cannot really feed themselves, and they cannot even breathe. And they have severe hypoglycemia, and that's the reason they die. But very little enzyme activity, up to 8 to 25%. And this occurs, certain mutations may decrease the enzyme activity, but they may not totally erase it. In this case, the patients survive adult age up to 30, 40 years of age, and disease occurs after them. And the reason that occurs, of course, is because polyglucosan accumulates in the neurons, and it causes it prevents the signal transduction between the brain and our muscles, or between the sensory receptors in our skin and the brain. So the patients really experience neuropathy or sensory neuropathy. And I copied this slide from APBD Research Foundation website. So it basically describes the APBD and among the rare diseases, its occurrence and how many patients are there. So they also summarize that there are more than 40 known pathogenic mutations in the gene that causes APBD. The most common one is the mutation that causes tyrosine to serine change in the gene. This mutation, due to founder effect, mostly found in Ashkenazi Jewish population. But I mean, our studies and the communication with the caregivers and patient, the doctors make us know that actually there are also other patients who are not Ashkenazi Jewish yet carries this mutation. In 2015, we have also described another mutation as common as this exonic mutation. It's an intronic mutation, which affects the messenger RNA synthesis and eliminates the enzymatic activity from the affected allele almost totally. And how do we diagnose the APBD? The APBD has been diagnosed by the blood test. In the blood test, we can do or genetic testing and enzymatic assay. And but nowadays, with the development of the DNA sequencing, and it is very common use in the diagnostic methods. Now, almost exclusively, the doctors or the centers use DNA sequencing. And if they find any other mutation other than 14-ol mutations, then we look at the enzyme assay and determine if that mutation causes APBD or any other disease. So basically, the mechanisms that causes the branching, poor branching, and the polyglucosan body, besides the GBE deficiency, is the PFK deficiency and the ubiquitin ligase deficiencies, which they increase the glycogen synthase activity compared to branching enzyme. And this slide summarizes how histologically this disease is being diagnosed. As you can see here, we have neuron axons, which are actually the wires of the communication wires of the cells, our body, that actually conduct the signal from brain to the neurons or from the sensory from our skin to the brain. And if, as you see, the polyglucosan body accumulates in the neurons and blocks the signal transduction. And in the muscle, you see here the polyglucosan precipitates in the muscle. And those muscle fibers that has this polyglucosan, of course, they cannot obtain enough energy to contract when they need it. And this diagram shows that the patients first experienced bladder dysfunction around 20, 30 years of age, and later disease progressed to difficulty in walking. They become wheelchair bound around 60 or 70 years of age. So is there any treatment for APBD? Currently, there is no treatment. Unfortunately, this disease has a very special, specific defect that causes the accumulation of polyglucosan in the cell. And this accumulation cannot be reversed because the polyglucosan, there is no enzyme that can digest the polyglucosan in our body. But we have some experimental methods or strategies that we are trying to employ and eliminate the disease. One is, of course, the gene therapy. Gene therapy means providing the cells with the gene that is defective. So, okay, patients don't have the GBE1 branching enzyme gene. We can give the gene back into the cell using the viruses. So that is called gene therapy. And in this way, actually, you can treat pretty much every genetic disease. But of course, right now, the technology, the viruses that we use allows only the small genes. And it's very fortunate that GBE1 is small enough to get into the virus and be delivered into the cells. The other approach is using the small molecules that prevents the glycogen synthesis. So the glycogen synthase activity is very high compared to branching enzyme activity because of mutation or some other reasons. So if we can decrease the glycogen synthase activity, we may provide enough branching, and we may, although it is going to be very small, but we are going to be able to produce normal glycogen. And then this is supposed to cure the disease. And the other approach is for a very particular mutation that we have described in Intron using antisense oligonucleotides. So antisense oligonucleotides are small stretch of DNAs. They are very specific only for that particular mutation. So not every APBD patient or glycogen storage disease patient can have this. But based on the particular mutation, it can be done. Especially intronic mutations are very theoretically easy to access and can be modified, so we can treat this disease using antisense oligonucleotides. So briefly, this particular mutation that we have described occurs in intron 15. So introns are it used to be called junk DNA. We have quite a lot of DNA in our body. They don't really code anything. At least we didn't know their function so far. That's why they use this term. I think it has been used quite wrongly, but there was no other way to describe, because this very long DNA sequences, they don't code any protein or enzyme, but it is just in between the DNA regions that you can see here, is just connect them. And the mutation that we have described is in intron 15 that actually prevents the synthesis of normal enzyme. So this mutation has been hidden for a long time because, first of all, it was in the intronic region. It is a lot bigger than the messenger RNA of branching enzyme itself. And that's why it was not being discovered before. But in the patients, they were who were heterozygous, as you can see here. Heterozygous means the patient have one gene from mother, one gene from father. And one of the gene that the patient inherited is normal. Here, the normal is supposed to be the green peak. But this green peak actually disappears in the messenger RNA, which shows that actually the patient do not use the gene from one of the parents that he or she inherited. Yet the gene that has been used is the mutated one. So, of course, the enzyme activity is very low, and the patient developed the disease. But why it didn't occur? So when we look at sequence the gene in the intronic region, we have found that the 9 base pair has been deleted, and there's a large insertion of 19 base pair into the intron. So, I mean, it looks like a small change, yet unfortunately, this insertion has a functional base, which changes the synthesis of the messenger RNA and, of course, the enzyme. Because of this change, as you see the structure here, this structure is not supposed to occur in this area. So because it occurred and it cut the message, it caused the disease. We can actually change this structure by using antisense oligonucleotides. They are a small stretch of molecules. As you can see here, they are like a zipper or the magnet. They bind to each other. So what we did with the Ionis pharmaceuticals, we designed specific small oligonucleotides, which make a zipper action and binds here before the DNA itself or RNA itself binds to that. So using this methodology or technology, we are able to recover this normal allele. And as you can see here, I am sorry, the slide is quite complicated, crowded, but here, when there is no oligo has been used, the mutated allele has been expressed. But in all other oligonucleotides that we have studied, you start to see the normal base, the green peak you can see here. So based on the degree that this antisense oligonucleotides binds and inhibits this formation, we recover the normal allele that the patient inherited from mother or father. And using this technology in the cells, we are able to also recover the enzymatic activity. So when there is no enzyme activity in the same cells, when we treat it with the particular oligonucleotides that we have used, as you can see, at certain concentrations, we got the maximum enzymatic activity. So this clearly shows that we can actually recover the normal gene in the patient, and then the patient is going to have normal branching enzyme activity that will eventually clear the polyglucosan in the cells. So right now we are at this stage, and I am collaborating with Dr. Hirano, and hopefully we are going to bring this treatment for patients using the with also the support of IMS Pharmaceuticals and NORAM Foundation. Before I end my talk, I would like to thank my collaborators, Dr. Salvatore Di Mauro, who's my mentor, Dr. Purnay Tanji, Dr. Hirano, and Valentin Emanuele for their support in this research and help. And we also have both financial and information. Actually, we got a lot of information from patients so that we could direct our research. I would like to thank APBD Research Foundation and Kate B. Hayes Foundation for their support, and IMS Pharmaceuticals. Thank you.